It's now my great honor to introduce our second speaker, Dr. Lori Passmore. So Lori graduated with an undergraduate degree from the University of British Columbia, and then from there she went on to do her PhD with David Barford in London at the Institute of Cancer Research, where she conducted structural and functional studies of the anaphase promoting complex. She then became a career development fellow at the MRC in Cambridge with Drs. Venki Ramakrishnan and Richard Henderson, where she conducted structural studies of complex involved in the initiation of eukaryotic translation. Following this, she became a group leader at the MRC in 2009, where her lab has made fundamental discoveries concerning the structure and function of the machines involved in regulating mRNA 3' ends, as well as the Faconia anemia complex. Dr. Passmore has received numerous awards and accolades, including the Suffrage Science Scheme Award, being elected to EMBO, and recently the inaugural Elisa Izaralda Award from the RNA Society. So to sum up, I'm really excited to hear Dr. Passmore's seminar today, titled Mechanistic Insight into mRNA Cleavage and Polyadenylation. So please join me in giving a warm, if virtual, welcome to Dr. Lori Passmore.
Thank you so much, Ben, for the introduction. And thank you so much for inviting me. It's a real pleasure to participate in the seminar series today. So I'd like to talk about a transcription-related process today, which is mRNA 3' end processing. And as you will all know, poly-A tails are found on almost every eukaryotic mRNA. And poly-A tails are added by a large multi-protein complex called CPF, or cleavage and polyadenylation factor. And that's CPSF in humans. So CPF is thought to travel along with RNA polymerase II, monitoring nascent transcripts for specific sequences. And when it recognizes those, it cleaves the pre-mRNA and it adds a poly-A tail. So it adds around 60 A's in yeast or around 250 A's in higher eukaryotes. But it's not only the length of the tail that's important, the position of the poly-A tail is also important. So a change in the polyadenylation site can result in either the inclusion or exclusion of regulatory regions from the 3' UTR of the mRNA. And this process is deregulated in disease. So for example, a single point mutation in one of the sequences of the globin gene can result in thalassemia. Polyadenylation is globally deregulated in cancer, and the machinery is also targeted by viruses. So poly-A tails are required for export of mRNAs from the nucleus. They bind to poly-A binding protein, and this is important for translation of mRNAs into proteins. Now, finally, poly-A tails are important for mRNA stability. And that's because the first and often rate-limiting step in mRNA decay is removal of the poly-A tail. So my lab's studying both how poly-A tails are added onto mRNAs and how they're removed. And these processes are all mediated by large multi-protein complexes. So over the past several years, we've developed methods to purify these, as you can see here. And we then use a combination of biochemical reconstitution and structural biology to gain mechanistic insights. And so today, I'd like to focus on the CPF complex that adds poly-A tails onto mRNAs. So this has 14 different subunits in yeast, and we can purify the native complex using a tap tagging method from yeast. The total mass of the complex is 850 kilodaltons, so almost 1 megadalton. Almost all of the subunits are essential for viability in yeast, but when we started working on this, there was really very little structural information on the complex. But I would say what really fascinates me about this complex is that it contains within it 4 different enzymes. So there's an endonuclease that cleaves the pre-mRNA, a poly-A polymerase that adds the poly-A tail, and there are 2 protein phosphatases that regulate transcription, and specifically transcription termination. So we really want to understand then how mRNA is recognized by CPF, how pre-mRNA cleavage is activated, how polyadenylation is regulated, and how this triggers transcription termination. So of course, because we could purify this complex from yeast, we tried to image this intact complex using Cryo-EM. But unfortunately, the complex dissociated when we made the Cryo-EM grids, despite it being very stable in solution. So we decided to do native mass spectrometry with Carol Robinson's lab to understand how all the subunits interact. So using native mass spectrometry, the entire complex is analyzed in the spectrometer, and ionization causes fragmentation of the complex into subcomplex. We can then determine the composition of these subcomplexes and build up an interaction map. So in total, we analyzed 38 different subcomplexes and used a computational network analysis to reveal the protein-protein interaction network within the complex. And so this turned out to be really interesting because it showed that the proteins of CPF associate via tight interactions within 3 different modules. And these modules are based around the enzymatic activities of the complex. So there's a nuclease module, a polymerase module, and a phosphatase module. Now, since we weren't able to obtain a structure of the intact complex, we decided to focus on one of the modules, and we started with the polymerase module. So there's 5 subunits in the polymerase module. All of these are conserved in humans. So here I've shown the human names in brackets, but I'll use the yeast names when I talk about them. So the largest subunit is CFT1. YTH1 contains 5 zinc fingers and was known to bind to the polyase signal of mRNA. PFS2 has a predicted WD40 beta propeller. FIP1 is a predicted unstructured protein, but it's known to bind to the polyA polymerase and PAP1 and tether it to the complex. So to study this, we decided to make the complex recombinantly, and we inserted the genes for all 5 of the subunits on a single baculovirus so that when we infected insect cells, they simultaneously produce all subunits and we can purify the complex now in milligram quantities. So this is work done by Ananth Kumar and Anna Casaniel. Here they are at the microscope, and they obtained these really beautiful micrographs of the complex. So each of these black objects on the micrograph is an individual polymerase module complex, but I should say that because the polymerase subunit itself tended to fall off the complex, we imaged the 4 subunit complex lacking the polymerase. So this is now around 200 kilodaltons. Now, if we take the individual particles from this micrograph and average those that seem to be in similar orientations on the grid, you can get these 2-dimensional class averages. And as you can see, there's a lot more detail now in these 2D class averages, and the detail that you see there is actually the secondary structure elements aligning. So together we could put all this data together and obtain a 3D reconstruction at around 3.5 angstroms resolution. Now, I want to point out that high resolution here is necessary because we didn't have any prior structural information. So we really needed to be able to read the sequence from the map itself. So at this resolution, you can clearly see alpha helices. Beta strands are well separated from each other. And as you can see, there's density for some side chains. On the right-hand side, there's a lower resolution part of the map where there's a zinc finger, and you can see the zinc being coordinated by the cysteines. Okay, so using these maps, we could build atomic models into the density for over 1,700 amino acids. And that gave us this model. Now, although our complex that we put on the grids contained 4 proteins, only 3 of them were visible in the map. So it's a relatively rigid structure, and at the bottom in green is the CFT1 subunit. It has a beta propeller right at the bottom here, a second beta propeller over on the side here, and a third beta propeller on the other side. There's a 4th beta propeller in the complex in the yellow subunit, PFS2, at the top. And then finally, the 3rd protein, YTH1, in magenta, interacts on the side of the complex, contacting both of the subunits. So I'm going to just very briefly go through some of the structural details. I promise it will be brief. So first of all, CFT1, the largest subunit in green at the bottom, starts over here. Its N-terminus is here in dark blue. And what you can see is it does a single strand of this beta propeller. It then comes over to the right-hand side here and completes the beta propeller on the right, comes back to the first beta propeller for 3 strands of this blade, then completes the bottom beta propeller, comes back to the first. And then finally, the C-terminal domain is this bundle of alpha helices here. So the way that these beta propellers are really intertwined means that they form this rigid structural core. And that's further stabilized by the alpha helical domain, which is sitting right at the nexus of the 3 beta propellers. So we think this really acts as a scaffold for the entire complex. Next, I'll show you YTH1 in magenta. So YTH1 actually has 5 zinc fingers, but we can only see 2 of them in our map. One of them is here, contacting both of the yellow and the green subunits. The other hangs off the side here. Now, YTH1 itself is actually anchored onto the complex by this long, extended N-terminal segment that binds right in the center of one of the beta propellers of CFT1. And so that's really how it's anchored onto the complex. And then finally, PFS2 buries 50 amino acids into the cavity that's formed by the 2 upper beta propellers of a CFT1. So this buries over 4,200 square angstroms of surface area. That's a lot. I think it really explains why we could never purify these proteins as individual monomeric proteins, because there's so much interaction between them. They're really an obligate complex. So in summary, we reconstituted this polymerase module of CPF. It's active in polyadenylation in vitro, and we determined a Cryo-EM structure of it. We were still missing, however, half of YTH1 and all of FIP1, which weren't visible in the structure. Now, we think this is because they're flexible and therefore they're blurred out in our maps. We know from other data that FIP1 binds to the polymerase subunit, PAP1. And in work I'm not going to talk about today from Ananth Kumar and Connie Yu, we used NMR to show that this, even in the entire 1 megadalton complex, this FIP1 really still does stay flexible. And so it may stay flexible to allow the polymerase to access the end of the RNA as it's polyadenylating it. But that work's posted on bioRxiv, so you can have a look at it there. So we also showed that the accessory factor, CF1A, that's required for cleavage and polyadenylation, interacts with the polymerase module directly. And both CF1A and the polymerase module interact with RNA. Now, work from Liang Tong's lab and Martin Ginek's labs have determined structures of the human complex bound to RNA, and that really nicely describes how the AAU-AAA sequence is recognized by this complex. However, we're still missing most of the CPF in our structures. So all of the subunits of the nuclease module and all of the subunits of the phosphatase module. And we really wanted to understand this, in particular the nuclease, because that's what really defines the 3' end of a transcript. So the nuclease subunit in yeast is called YSH1. And it contains a C-terminal domain that has low complexity regions. And at the N-terminus, it is the nuclease domain. So a crystal structure from Liang Tong's lab, determined in 2006, showed that the nuclease domain is comprised of a metallobeta-lactamase domain and a beta-casp domain with zinc-coordinated active site residues right at their interface. Now, in this structure, the nuclease is actually in a closed conformation. So the 2 domains are kind of pressed up against each other, and there's really no clear path for substrate RNA to enter the active site. Now, that's consistent with the fact that whenever we purify this protein, it has very weak and non-specific activity. So what we think this is telling us is that really an activation step is required for efficient and specific cleavage of pre-mRNAs. And that makes sense because you don't want a nuclease running around the nucleus or cytoplasm cleaving mRNAs non-specifically. So to understand how this nuclease is active, we started looking at the nuclease module. And this is work done by Chris Hale, a former postdoc in the lab. Now, Chris was able to purify a dimeric complex of the nuclease, YSH1, bound to another subunit called MPE1. And he could define that N-terminal ubiquitin-like domain of MPE1 binds to the YSH1. He determined a crystal structure, which really nicely defined the interface between them. And if you look at a cut through of the structure, you can see that the MP1 ubiquitin-like domain binds to the nuclease domain right next to the active site tunnel opening. And again, the catalytic core is inside this long solvent-filled cavity, right at the boundary between the two domains of the nuclease. And there's really just a narrow tunnel leading to the active site. Now, if we turn this sideways, you can see there's a long blue region here. This is a basic patch formed by contiguous surfaces of both YSH1 and MPE1, suggesting that this may be the path that RNA takes to the active site. However, the active site is simply too narrow to accommodate the RNA. So again, this nuclease is in an inactive conformation. So we decided to turn to biochemistry to see if we could figure out what was required for activation of the nuclease. And so to do this, we use the 3' UTR of a transcript from yeast, followed by the downstream region. And we would expect CPF to cleave at really quite a specific place. And then it could add a poly-A tail onto the 3' end of the 5' product. Now, the polyadenylation signal sequence, or PAS, is typically an AAU-AAA sequence. It's more degenerate in yeast, but AAU-AAA is still a commonly found PAS sequence. And this is what is actually recognized by the YTH zinc fingers. And that will become important in a couple slides. Okay, so to look at activation of cleavage, we didn't need to look at polyadenylation. We just studied the cleavage part of this reaction. And Chris was able to purify now a larger complex that contains all of the subunits of the nuclease and polymerase modules. So this is now half a megadalton in size. It has 8 subunits. And so when he incubated, we call this the core CPF. The core CPF, or smaller complexes that contain the nuclease with that RNA substrate, he found that the only thing that was active was the core CPF. And in fact, it was only active when we included the accessory proteins CF1A and CF1B. So this, I guess, really showed why it had been so difficult to reconstitute this reaction with recombinant proteins, because really you needed so many proteins in order to activate the nuclease. So now that we had an active complex, we went back to the microscope. And again, using Cryo-EM, the sample fell apart, but we were fortunate to get some images using negative stain, and these looked reasonably good. And so we could build a really low resolution model of the entire core CPF complex, and then we could fit in models that were known from X-ray or Cryo-EM structures into the map. And so what you can see is that on the left is the polymerase module, which I showed you in earlier slides, and on the right is the polymerase subunit, PAP1, as well as the nuclease module subunits. Now, what was somewhat surprising to us about this model was that the RNA binding site, the nuclease active site, and the polymerase active site were not in intimate contact with each other. We thus hypothesized that CPF really may be much more dynamic than we previously thought, such that RNA binding would activate a series of conformational changes to activate the nuclease and to regulate polyadenylation. Excuse me. So we think maybe the coupling between the different enzymes may be dynamic and regulated. Okay, so to try and get insight into how RNA binding might activate the nuclease, we decided to study the orange subunit here, MPE1. And this is work done by Juan Rodriguez in the lab. So MPE1 is a subunit of the nuclease module. It's essential for viability, and it's known to stimulate cleavage and polyadenylation in vitro. MPE1 is a multi-domain protein. You can see the domain diagram along the bottom of the slide here. It has an N-terminal ubiquitin-like domain, which I showed you on the previous slide, that interacts with the nuclease directly. It has a central zinc knuckle, and it has a ring finger that's towards the C terminus. Now, there's some evidence that MPE1 directly interacts with RNA. So Juan started studying this, and he found that MPE1 interacts directly with the polymerase module, and this interaction is stabilized by RNA. So he took this complex and he studied it by Cryo-EM and obtained a map at 2.7 angstroms resolution. So in this map, we identified additional density, it's shown here in orange, that wasn't present in any previous maps of the polymerase module. So this new density extends here from the top of PFS2 down the side, reaching right into that intersection where PFS2, YTH1, and CFT1 all intersect. So given the high resolution of this map, again, we could de novo build an atomic model into that new part of the density. And that looked like this. So there's a kind of compact folded region at the N-terminal part here, and then it extends down into a helix. And that actually maps to the linker region that is between the zinc knuckle and the ring finger of MPE1. Now, Juan had also included RNA in his sample. And so we also identified in gray over here, some density that corresponds to 3 nucleotides of the polyadenylation signal of the RNA. And so, as you'll see in a minute, we now refer to this region of MPE1 that binds to the polymerase module as the PAS sensing region, or the PSR. Okay, so let's look at the RNA in a little bit more detail. As I said, we see three nucleotides. And the first thing I would say is that the molecular mechanism of how the A1 and A2 of the PAS is recognized is highly conserved with human. It's almost identical to the structure of the human complex. However, we also see that there's one nucleotide upstream of the PAS that's well ordered. This wasn't in any of the previous structure. It's a U in our case, and it's recognized really in a surface pocket of the zinc finger protein YTH1. So we think this may explain how the context of the PAS sequence may contribute to its recognition. But more surprisingly, we found that the A2 of the PAS is contacted directly by MPE1. And specifically, this proline 215 forms a CH pi interaction with the A2. Now, in MPE1 homologs, proline 215 and the surrounding residues are all highly conserved, suggesting that PAS binding by MPE1 may be conserved across all eukaryotes. So based on this structure and the data that I'll show you in a minute, we hypothesize that MPE1 is really sensing when the polymerase module has bound to the correct RNA. But to test that, we needed a lot of CPF. So Juan used a system developed by Anant in the lab where he could co-express all 14 genes off of 2 BACmids in baculovirus. And this allows him to overexpress all 14 subunits and ultimately to purify the entire 1 megadalton complex as a recombinant protein. So this was really exciting because we could now use this to study the role of MPE1 within the intact complex. So first of all, we wanted to understand the role of MPE1 in cleavage. And here we used a short synthetic RNA, and it has different fluorophores at the 5' and 3' ends. So here you can see with wild type CPF, the intact RNA has both fluorophores. It's in purple at the top. When it's cleaved, you get the red 5' product and the blue 3' product. So if Juan makes CPF lacking MPE1, he found that it was actually 10 times slower at cleaving the RNA substrate. So MPE1 seems to be a major activator of cleavage. Now, interestingly, you'll notice next to this asterisk, there is an increased cleavage at alternative sites when MPE1 is missing. So this is perhaps consistent, we think, with the MPE1 playing a role in actually selecting the cleavage site by contacting the PAS in the RNA. Now, Juan also made a mutant in MPE1 that would disrupt the direct contact with A2 of the PAS. So he mutated the proline 215 to a glycine. And when he did that, cleavage again was very severely disrupted. So this is a single point mutation in MPE1. Now, interestingly, this single point mutation caused a more severe defect in cleavage compared to the complete loss of MPE1 above it, really suggesting that this defective MPE1 may be preventing other subunits of CPF or CF1A or CF1B from compensating for the lack of MPE1. So what about polyadenylation? We did the same experiments with polyadenylation. Here, the RNA is at the bottom of the gel, and over time, CPF adds adenosines, making a poly A tail. And so what I hope you can see, although it's a little bit subtle, I think it's very reproducible. When CPF is lacking MPE1, its polyadenylation activity is reduced. So it's more slowly adding the tails onto RNA. But what was the big surprise was when we used this proline mutant, CPF hyperadenylated the RNA substrate. So now all of the RNA is gone up very quickly to the top of a gel and is running in the wells. So MPE1 is really required for regulated polyadenylation. So the last thing I want to show you is some experiments that we did to understand the role of MPE1 in vivo. So we investigated the consequence of acute depletion of MPE1 on the yeast transcriptome. And we did this by using a mini auxin-induced degron at the C-terminal end of the endogenous MPE1 gene. And as you can see here from this Western blot, when we add auxin, depletion of MPE1 occurs within 30 minutes. So we did this and we sequenced nascent RNA after MPE1 depletion and compared this to control cells. And I don't have a lot of time to talk about this. I'll just say that when we deplete MPE1, it causes a major significant change in the nascent RNA levels of over 2,600 genes. And very specifically, it led to a widespread increase in the signal downstream of the polyadenylation sites, shown here in pink. So this is a metagene plot of all transcripts aligned at the poly A signal. And normally, after cleavage, the RNA is degraded that's downstream of the poly A signal, as you can see in the purple control. But in pink, you see it's not degraded at all, or there's still a lot of signal. So these results are really consistent with defects in 3' end processing and transcription termination. And they're also indicative of RNA polymerase II read through beyond the normal 3' end of the transcript. And in fact, this defect is as severe as if you deplete YSH1, the nuclease, from the nucleus. Okay, so just in summary, I'd like to present our current model. Now, I would say that in other studies, it has been suggested that the ApoCPF complex, in the ApoCPF complex, the endonuclease is simply too far away from the RNA. And it would require a spatial rearrangement in order to bring it in close proximity with the RNA and an activation step. So here we show that we think MPE1 plays a major role in mediating this. So we hypothesize that RNA recognition by CPF and the accessory factors CF1A and CF1B promotes a series of conformational changes that activate pre-mRNA cleavage and transcription termination. So we've shown that the MPE1 subunit directly contacts the polyadenylation signal sequence in RNA, and this promotes the structural rearrangement that would probably prime the activation of the CPF endonuclease and control the poly A polymerase activity. So in vivo, depletion of MPE1 leads to widespread defects of transcription termination by RNA polymerase II. And so overall, we think MPE1 enables activation of the endonuclease activity. It enables controlled polyadenylation and transcription termination. We think it's possible that assembly of the 3' end processing machinery may be analogous to the assembly of an active spliceosome, whereby it only becomes activated when the correct RNA sequence and all the correct accessory factors are assembled. And this would really ensure fidelity in 3' end processing. And so just to finish off, I'll thank the people who were involved. The MPE1 work is unpublished. It was all done by Juan in the lab. Polymerase module work was done by Anna and Anand. Chris Hill worked on the YSH1 aspect. Yshl Easter started out a lot of the work on a CPF complex in my lab 10 years ago. I think I've mentioned all of our collaborators as we went along. I'm thankful to all of these people for funding and happy to answer any questions.
Thank you so much. That was just beautiful. I'm always amazed when you can actually sequence the sequence of the RNA by the structure. Just incredible. So there's a few questions already. So Masahiro Nishimura says, thank you for your impressive talk. I would like to hear if you considered that the structural model of CPF can be improved by AlphaFold predictions.
Yeah, so I guess looking at the structures of really big dynamic multi-protein complexes is still difficult, but we've looked at some of the parts of the complex, and certainly we have some new predictions that we can have a look at. So yeah, it's exciting. It's an exciting time, and we're really looking forward to looking at that more in detail.
Awesome. And then an anonymous question which says, "Great talk!" exclamation mark. As polyadenylation is deregulated in cancer, are there any indications that human MPE1 is mutated or altered in human malignancies, or are there other subunits that are currently mutated to allow alternative polyadenylation, given the tight control of endonuclease activation?
Yeah, that's a really good question. I haven't looked for mutations in the ortholog. It's called RBBP6 in humans. It interacts with P53 and PRB, which are, as you know, involved in cancer. So it's certainly possible that MPE1 is directly deregulated. There is something that happens, is that MPE1 in humans has two isoforms, and one of them, let me get this right, has only the ubiquitin-like domain, and so it inhibits the complex. So certainly there is some regulation happening there that we don't understand, and yeah, it would definitely be interesting to look at that in more detail.
Yes, and back to yeast, but on the same lines, have you tried making the MPE1, the proline point mutant to glycine in yeast, and is that viable and have interesting phenotypes?
We haven't tried doing that. It would be an interesting experiment. Yeah.
And then there's another talk from Anirban Dasgupta, who says, fabulous work. Apart from MPE1, do you think the polymerase complex contributes to the specificity of the cleavage process?
Yeah, so I guess that's what I was, yeah. So I think that there are many different parts of the complex that recognize different parts of the pre-mRNA. So I talked a lot about the polyadenylation signal sequence, but there's upstream sequences and downstream sequences. They're reasonably degenerate, but they have specific binding partners. And we think that all of the binding partners, including CF1A, CF1B, and possibly other parts of the complex, have to bind to all of them simultaneously. And that's what enforces the specificity.
Great. And then one more question from Laurent Boulanger, who says, do you think that one day it'd be possible to manipulate these complexes to make them cut RNA anywhere on a specific site?
Interesting. It's difficult because I think this particular complex is really, evolved to specifically recognize certain sequences. If I was to do that, I probably wouldn't choose the CPF to do that with, because I think there are too many things you would have to change, and it's not, there are too many sequences required for site-specific cleavage.
Yeah, I was amazed in your talk just how complex it is, the sequence recognition with all the different parts of different proteins. Okay, so I think we'll wrap it up there. So thank you, Lori, and thank you, Yasuhiro, for both of your fantastic seminars today.